Okay, so welcome back to this next video in which we are discussing endothelium-dependent vasodilatation. Okay, so we've seen how the arterioles control how much blood flows to a certain area, okay, and that the way in which arteriole diameter is controlled is by those vascular smooth muscle cells which line the arterioles. Now, all the time, the arterioles are receiving contraction promoting stimuli, okay, which we've seen can either open ligand-gated ion channels leading to depolarization and the opening of voltage-gated calcium channels, or can activate G-protein coupled receptors and mobilize calcium from the sarcoplasmic reticulum via the GQ/11 pathway, which we've just discussed. Okay, and then that calcium signal is promoting the contraction of the smooth muscle cells. Okay, what we now want to discuss is the other side, the thing that is counterbalancing this pro-contraction stimulus and is keeping the arterioles at a normal size, okay, which is the stimuli coming from the endothelium. Okay, so we're now going to look at the mechanisms of endothelium-dependent relaxation. Okay, right. So firstly, what we need to do is discuss what fundamental signal triggers the endothelial cells to start sending all of these different signals to the vascular smooth muscle cells. Okay, and again, it's a calcium signal. So if I now draw an endothelial cell here, so here is our endothelial cell, here is its nucleus here, okay. Basically, it is calcium going up inside these endothelial cells that causes the endothelial cells to start sending signals to the vascular smooth muscle cells that they need to relax, basically. Okay, now what causes calcium to go up in the endothelial cells under normal circumstances, and therefore what drives the normal signaling between the endothelial cells and the smooth muscle cells? Well, it's normally shear stress that drives the rise in intracellular calcium that causes the normal homeostatic signaling between the endothelial cells and the smooth muscle cells. So what does shear stress actually mean? Well, remember these endothelial cells, they are lining the lumen of the blood vessels. Again, we're talking now not just about the arterioles, but also the other microcirculation vessels as well, the capillaries and the venules. Blood is going to be flowing over them. Okay, there is going to be a friction of the blood against the smooth muscle cells. Okay, so molecules are going to be ramming onto molecules that are on, sorry, I said smooth muscle cell, I meant endothelial cell. Molecules in the blood are going to be ramming into molecules that make up the endothelial cell apical membrane, and therefore this is going to be having a friction effect, basically. And that's what is meant by shear stress, this force that is on the surface of the endothelial cells that is generated by the movement of the blood over their surface. Okay, and through mechanisms that are still not quite understood, this leads to rises in intracellular calcium within the endothelial cells. Okay, so it causes calcium signals within the endothelial cells, and these calcium signals, as we're going to see, are responsible for initiating all three of the different aspects of endothelial dependent relaxation. Okay, right. In addition, under the circumstances of inflammation now, this is not under normal homeostasis, we're now talking about inflammation. Under inflammation circumstances, what's going to happen is certain inflammatory mediators are going to activate receptors on the surface of the endothelial cells that then cause rises in this calcium signal within the endothelial cells and therefore promote endothelium-dependent relaxation. And that's what leads to the vasodilatation of the arterioles in the inflammatory response that increases the amount of blood flowing to an area that is undergoing an inflammatory response. Okay, so key inflammatory mediators that are responsible for this are histamine, okay, and also bradykinin. Okay, so let me just talk about these a little bit more. Histamine and bradykinin both have G-protein coupled receptors on the surface of the endothelial cells, which are GQ coupled G-protein coupled receptors. So the G-protein coupled receptor for histamine that's on the surface of these endothelial cells is a receptor known as H1. Okay, the G protein coupled receptor on the endothelial cells for bradykinin is known as B2. Okay, so let me just draw these on here. So here is our G protein coupled receptor, and these are going to be GQ coupled G protein coupled receptors. Okay, so when you activate these receptors on the surface of the endothelial cells, that is going to activate the calcium signaling that we have just discussed in the smooth muscle cells, which is going to result in the release of calcium from the endoplasmic reticulum within the endothelial cells. Okay, and that's going to raise calcium in the cytoplasm of the endothelial cells, and that's going to bump up endothelium-dependent relaxation signals, and therefore they're now going to overwhelm the contraction signals, and therefore you're going to get net dilatation, relaxation of the vascular smooth muscle cells, which then leads to the increase in the diameter of those arterioles and an increase in the blood flowing to that inflamed area. Okay, right, so that's how these inflammatory mediators, histamine and bradykinin, can increase endothelium-dependent relaxation. So now what we want to look at is how does this calcium signal cause the three different components of endothelium-dependent relaxation. Okay, and the first one that we're going to look at is nitric oxide. How does it trigger the generation of nitric oxide? Now I want to stress a point that I have stressed before, but I'm going to do it again, which is that these mechanisms are going to be occurring in not just the endothelial cells of the arterioles, but also the endothelial cells of the capillaries and the venules in this local piece of tissue, and they're all going to be sending nitric oxide to the vascular smooth muscle cells surrounding the arteriole over here. Okay, right, so let's talk about how these endothelial cells then are going to generate nitric oxide. So firstly, let me show you the structure of the molecule nitric oxide, and then we'll discuss how you're going to activate the production of nitric oxide by a certain enzyme. Okay, so nitric oxide, often just abbreviated down to NO for short, and it also has another name, which is known as the endothelium-derived relaxation factor, okay, EDRF. So this is endothelium-derived relaxation factor. Now, as we'll see later on, there are actually other factors that are released by the endothelium that produce relaxation of vascular smooth muscle cells, but nitric oxide is most likely the most important one, and therefore it's the one that's named the endothelium-derived relaxation factor. Okay, so when people talk about the endothelium-derived relaxation factor, generally that's synonymous with nitric oxide. Okay, so EDRF. Right, let me just talk about its chemical structure then. It's got a very simple chemical structure. It is literally just a nitrogen atom bound by a double bond to an oxygen atom. Now that's a brilliant arrangement for the oxygen, because oxygen only needs two bonds. Okay, however, it's not such a good arrangement for the nitrogen, because nitrogen is most comfortable with 3 covalent bonds, and in this arrangement it's only got 2 covalent bonds. Now that actually means that one of the nitrogen's electrons is sitting on its own, okay, which we'll show like this. Okay, so it's not in a pair. Okay, it's not paired, it's on its own, which means that nitric oxide is actually what's known as a free radical. So chemical structures which contain unpaired electrons are known as free radicals. Now, free radicals are usually seen as very, very dangerous in biology, okay, because they are incredibly reactive generally, okay. Even the general public know that free radicals are not good. They are very, very famous, okay. However, nitric oxide is actually a very stable free radical, okay, so it's not too bad. So don't be scared by this molecule, it's not as bad as, say, superoxide. Okay, right, so it is a free radical and that's its structure. Right, so what I now want to discuss is how you are going to produce nitric oxide. Now, before I just say that, let's put the link between how calcium is going to activate the enzyme responsible for nitric oxide in first. Okay, so what I'm going to say just before we discuss the structure of the enzyme then is that calcium, when you generate this calcium signal inside the endothelial cells, what that is going to do is it's going to bind to calmodulin molecules, and again you're going to get the formation of calcium calmodulin complexes within the endothelial cells. Okay, so it's the calcium calmodulin complexes that are going to be responsible for activating the enzyme that is involved in producing nitric oxide within these endothelial cells. So here is our calcium calmodulin complex here. Okay, right. And what's it going to activate? Well, let's now finally discuss the name, at least, of this enzyme that's going to produce nitric oxide. Okay, so the enzyme that produces nitric oxide in the endothelial cells is called eNOS, okay, and it has another name as well. It's also known as NOS3. Okay, NOS3 is the slightly more correct name. Okay, so what does NOS stand for? Well, NOS stands for nitric, that's the N, oxide, that's the O, and then the S is for synthase, so it's nitric oxide synthase. So NOS3 stands for nitric oxide synthase 3. eNOS stands for endothelial nitric oxide synthase. Okay, because the main place where nitric oxide synthase 3 is found is the endothelium, and therefore you can call it the endothelial nitric oxide synthase. Okay, and it certainly is the main form of nitric oxide synthase that is found in endothelial cells. There are other forms of nitric oxide synthase in the human body. There is NOS1 and NOS2 as well, okay, but those have different roles elsewhere. Okay, so eNOS is the main nitric oxide synthase enzyme in endothelial cells, okay, and it's going to be activated by calcium calmodulin complexes. Right, so before we actually discuss the activation of eNOS by calcium calmodulin complexes, what I firstly want to discuss is the reaction that the eNOS is actually going to catalyze, and this is actually quite complicated. So we're going to start off by discussing the actual reaction by which you produce nitric oxide synthase, and then we're going to actually discuss the structure of this enzyme and how calcium calmodulin complexes are going to activate it. Okay, so let's start off then with the actual reaction that it's going to catalyze. So nitric oxide then is produced from the starting material of an amino acid. Okay, so the starting material is actually L-Arginine, which is an amino acid. Okay, so I'm going to start off then by showing you the structure of L-Arginine. Now firstly, let's just get over the L here. The L just refers to the specific optical isomer of arginine. So remember, amino acids have two optical isomers, the LAVO-rotatory, the L isomer, and then the dextro-rotatory D isomer. Now in biology, it's always the L isomers that are found within cells. Okay, so we're not going to dwell on that point too much. Now, I'm going to draw the core amino acid structure in this first diagram, but later on I'm not going to bother drawing the core amino acid structure because this isn't going to be affected by the reaction. Okay, so here is the core amino acid structure here, like so. So we've got the alpha carbon here, the amino group here, the carboxylic acid group here. This is the same structure as you have in all amino acids. Okay, all proteinaergic amino acids anyway. Now the R group of L-arginine is what we're interested in, and after this picture, this is all I'm actually going to draw of the arginine. Okay, the R group of L-arginine contains 3 methylene groups, okay, and because I want this to fit nicely onto the page, I'm going to just draw 1 methylene group and put brackets here, and then subscript it 3, which means copy this out 3 times if you can be bothered. Okay, but that denotes that there are 3 methylene groups. And then following that, you then have a nitrogen with a hydrogen coming off it. And following that, you have a carbon which then has an amino group coming off here. And it's also double bound to a nitrogen atom here, which then has a hydrogen coming off it. Now, this nitrogen, which this carbon is double bound to, has a very special name. Okay, so this nitrogen where the carbon's double bound to it, this is known as the guanidino nitrogen. Okay, and that's going to be important later on when we come to name the next intermediate in this reaction. Okay, so that's the guanidino nitrogen. I'll just underline it. So that's the structure of L-Arginine. Now, of course, the L is just going to refer to the specific optical isomerism of this alpha carbon here, because this is a chiral center with 4 separate groups coming off it. Okay, so we're not going to dwell on that. Right. So, what's now going to happen then is the endothelial nitric oxide synthase is going to catalyze 2 steps of this reaction, okay? So the synthesis of nitric oxide can be split into 2 steps, and both of them are going to be catalyzed by the endothelial nitric oxide synthase. So I'm going to show you these 2 steps now. And as I say, I'm going to cut out this portion from the pictures after now. So all of this is going, and I'm just going to show the R groups, okay, because the reaction is all about the R groups. Okay, right. So what's going to happen then first is that this arginine R group is going to be transformed, okay, so that off this guanidino-nitrogen, you now actually have an alcohol group rather than a hydrogen. So let me show you the end product of this reaction before I actually show you how it occurs. Okay, so the 3 methylene groups here of this R group have not changed at all. Here then is that amino group there. Here's the carbon. This amino group again has not changed. And now here we have the guanidino-nitrogen that now has this alcohol group coming off it here. Okay, so what's this molecule called then, this amino acid? This is now called Ng, okay, which stands for guanidino-nitrogen. The G is for guanidino, the N is for nitrogen. And then it's Ng-hydroxy, because you've got a hydroxy group coming off the guanidino-nitrogen. And then it's still L-Arginine. So it's just L-Arginine where you've got a alcohol group coming off the guanidino-nitrogen. So that's the name of this molecule. Okay, and remember, this core structure would still be there in reality. Okay, so how is this reaction achieved then? What do you have to put in to do this reaction? Well, you have to bring in a molecule of NADPH, okay, reduced NADP. Okay, so let me just explain something about this molecule. Okay, so NADP, nicotinamide adenine dinucleotide phosphate in full, so I'll write that out at least once. So the N stands for nicotinamide, okay, then the A is for adenine, and finally, well, sorry, not finally, penultimately, the D is for dinucleotide, and then the P is for phosphate. Okay, now, this molecule usually has a positive charge. Okay, so it's usually a positively charged molecule. Now, what can happen is this molecule can accept a hydride anion. Okay, so let me just explain what a hydride anion is. A hydride anion is quite rare. Usually we talk about hydrogen atoms which have become ionized by losing an electron. A hydride anion is a hydrogen atom which has become ionized by gaining an electron. So effectively, it is a proton at the center with 2 electrons orbiting it in a pair. Okay, a hydrogen atom that has gained an additional electron. So let me just cover this in. So remember, normal hydrogen just consists of 1 proton at the center of the nucleus with 2 electrons orbiting it. Okay, and no neutrons. At least that's the normal isotope of hydrogen. Okay, so this molecule can accept a hydride anion, and then it becomes what is known as NADPH. Okay, now, we view NADP as an electron carrier. Okay, it is carrying 2 electrons in the form of this molecule. We do not care about the proton. Okay, the important thing that it carries is the electrons. Now, why do we not care about the proton? Well, because protons are free in solution. Okay, in a solution you will have protons free that can be used in chemical reactions. Okay, protons are not difficult to come by. Electrons are more difficult to come by. So, effectively, we're only concerned about the fact that this carries 2 electrons. You can think of it, if you like, as carrying 2 hydrogen atoms, because when this is involved in this reaction, it's going to give the 2 electrons into this reaction, and then what will happen is you'll take 2 protons from solution, and therefore overall the molecule is going to receive 2 hydrogen atoms, because you've got 2 protons from solution, 2 electrons from this electron carrier, and therefore overall that's 2 separate hydrogen atoms, okay, that are being put into this reaction. But the way you should think of NADPH is that it is just carrying the electrons. It's providing the electrons. Don't get fussed up about the fact that it also has that proton there. Just remember which electrons is it bringing, and then concentrate on the fact that you can just get protons back to balance the charge of the electrons and make up the full hydrogen atoms from free solution. So the best way to view it, as I say, is just to view it as carrying 2 electrons, and then you can get 2 protons to pair with those to make 2 hydrogen atoms from free solution. Okay, so you're going to bring in this NADPH, which is going to provide 2 electrons, okay, into this reaction, okay, and then the 2 protons, you're going to get those from solution. Okay, and that other proton that was attached to the NADPH, that will go off into solution, and it could, if you like, if you want to imagine, it could be the one that ends up coming into the reaction from solution. Okay, but when we see the enzyme, you'll see that all this actually provides is the electrons. The proton gets chucked off. Okay, right. So NADPH is bringing in 2 electrons, which we're going to get 2 protons from solution to pair with. So effectively we have 2 hydrogen atoms there. The other thing we're going to need to bring in is oxygen, obviously, because where else are we going to get this oxygen from? So this is a dioxygen molecule. It's 2 oxygen atoms bound together. So I'm not going to show you the electronic flow diagram for this reaction. I don't know what the electronic flow diagram for this reaction is. What I'm going to show you is how it makes sense, okay? So what you can imagine doing is splitting the double bonds between those 2 oxygen atoms and creating 2 oxygen atoms back again. So give 2 of the electrons from this double bond back to this oxygen atom and the other 2 electrons back to this oxygen atom, and you then create 2 pure oxygen atoms back again. Okay, then what you can imagine doing is breaking this bond here between this nitrogen atom and this hydrogen atom. Okay, give 1 electron back to the nitrogen and 1 back to the hydrogen, and then take one of the oxygen atoms here and stick it in between them. Okay, so reforge a bond between this nitrogen and this oxygen, and then a bond between the oxygen and the hydrogen, and that creates your molecule here, okay, the Ng-hydroxy L-Arginine. Okay, now, finally, what's going to happen to the other oxygen? We need to get rid of that other oxygen. Well, what's going to happen is that other oxygen is then going to bind to the 2 hydrogen atoms that we've got by bringing in these 2 electrons and these 2 protons, and that then is going to create us a water molecule, H2O. Okay, so that's overall the reaction here. You've got 2 electrons from the NADPH, the reduced nicotinamide adenine dinucleotide phosphate there. Okay, so that's the first stage of the reaction, and at the moment you will notice that we have not yet created nitric oxide. Fail. Okay, so we need to now go on to the second reaction to see the creation of nitric oxide. Okay, so once again, what I'm going to do is I'm going to show you the final end point for the amino acid before I actually talk about the reaction. Okay, so once again, the free methylene groups are not going to be involved here. Okay, so the free methylene groups are left well alone. Here is the amino group again, that's been left alone. The carbon with this amino group coming off, that's been left alone. But now this carbon has been bound to this oxygen by a double bond. Okay, now this molecule here, this is what's known as L, because remember we've still got the chiral center back here in the L form. Okay, this is known as L-citrulline. Okay, right, and that's the final product in terms of the amino acid that you're getting from this reaction. Okay, right, now, what then are you going to do? Well, again, what you're going to do in this case is you're going to put in half a molecule of reduced NADP. Okay, now that's going to be providing 1 electron into this. Okay, so only 1 electron is coming this time. Half a molecule is being donated in. So 1 electron, and then of course you'll also get 1 proton from free solution. Okay, now what it's going to generate, and I'm going to go over what it generates and what we put in, and then we'll have a look at why this makes sense before we actually, so I'm going to tell you what the products and the reactants are before I tell you what the actual steps are. Okay, and then the other thing that you're going to produce is nitric oxide here. And the other thing you have to put in, and I'm sorry I'm jumping between reactants and products here, is an oxygen molecule again, a dioxygen molecule. Okay, so now let's go through what we're going to do here. Oh, and the other thing you're going to get out, I'm really sorry for this, is another water molecule. Okay, so what have we done here then? Well, what you can imagine doing, let's firstly think about how we can create the nitric oxide from this molecule here. Okay, what you can imagine doing is breaking that double bond there between this carbon and this nitrogen atom. Okay, give 2 electrons back to the carbon, 2 back to the nitrogen. Okay, then break this single bond between the oxygen and the hydrogen. That will spit off a hydrogen atom, so we've got a hydrogen atom to use up later on. Now, this oxygen has a free electron, this nitrogen now has 2 free electrons. What you're going to do is bind one of the free electrons of this nitrogen to the free electron of that oxygen. That will create you nitric oxide, okay, and the final free electron of the nitrogen will be unpaired. Okay, so that's our nitric oxide molecule produced. Okay, right. Now we need to clear up this mess. So we've got the nitric oxide. Okay, what you can also imagine doing is breaking the double bond between this dioxygen molecule again, okay, to create 2 oxygen atoms. And what you can do is bind one of those oxygen atoms, the 2 free electrons that that oxygen atom has, to the 2 free electrons that this carbon has after we split this double bond here, and that will create you the L-citrulline molecule. So we've got the L-citrulline, we've got the nitric oxide, we've used up one of these oxygen atoms here. Okay, now what we have to do is clear up the other oxygen atom. We have to clear up this hydrogen atom, and oh look, we've got another hydrogen atom coming in courtesy of our electron donor here, so we can then just create water with what's left. Okay, so that's how the reaction makes sense. Again, that is not an electron flow diagram. Okay, so that is how you produce nitric oxide by the conversion of L-Arginine into L-citrulline, and that's the reaction that the nitric oxide synthase enzyme is going to be catalyzing. Okay, right, so what I now want to show you is the structure of nitric oxide synthase 3, and in fact this structure that I'm going to show you, it goes for not just nitric oxide synthase 3 but also nitric oxide synthase 1 and nitric oxide synthase 2. Okay, right, so the first thing to say is that nitric oxide synthase 3 proteins on their own as a monomer are not functional, even though they have all of the domains they need to function. Okay, now this is a difficult, subtle thing to understand, and you're only going to understand it once I've actually shown you how this works. Okay, so to function, nitric oxide synthase 3 needs to work as a homodimer. Okay, so homo means same, dimer means 2-membered structure. So this is 2 identical nitric oxide synthase 3 proteins bound together to form a homodimer. So let me now show you the structure of this. So I'll start by showing you one nitric oxide synthase 3 protein, and then I'll dimerize them together. Okay, and initially I'm going to show you it in the inactive state, and then we will move to showing it in the active state. Okay, and I've just realized I haven't really left enough space here. Okay, never mind, we'll just have to make do. Okay, so here is the amino terminus of one nitric oxide synthase 3 protein. You then have a little linker here. Okay, then another important domain that I'll have in a box here. And then another important domain after that one, which I'll have here. Okay, right, and then here is the C-terminus. Right, so the 2 fundamental domains which we can divide up are this one here, which is at the amino terminus, okay, and then this one back here, which is closer to the C-terminus, which is in green here. Okay, now this one in purple, this is known as the oxygenase domain of the nitric oxide synthase 3 protein. So this is the oxygenase domain. Okay, whilst this domain here, this is known as the reductase domain of the nitric oxide synthase 3. So this is the reductase domain. Okay, right. Then you're also going to have another one of these dimerized with this nitric oxide synthase free. Okay, so dimerized with this, you're going to have another nitric oxide synthase free. Okay, I'm going to have to call it there for this video. We will come back to this in the next video.